All right, welcome back. So today we're going to talk about linkage. Linkage is along the lines of what we've been talking about recently, which is things that Mendel didn't run into. So when he was coming up with his law of independent assortment, he did not have to deal with any sort of linkage. Linkage is when you have genes on the same chromosome so that they travel together through meiosis and end up in the same gametes. And then we'll also talk about how they can be, so genes that are linked can be separated, and that's via recombination, so homologous recombination. And then lastly, what we'll do is we'll talk about how to actually map where genes are located on chromosomes relative to one another. So this was the early mapping based off of linkage analysis. This was not, a time at which we could actually sequence DNA. So before we could sequence DNA, we would have a general idea of where genes were relative to other genes. And so that would be mapping. Now, before we begin, let's do a little review so that we're all on the same page. So on the title slide, I mentioned that this is something that Mendel didn't run into, right? So let's talk about what Mendel saw and what we've learned as Mendelian dominance and classical Mendelian genetics. So basically what we're talking about is the principal segregation. The principal segregation states that diploid organisms have two homologous alleles. They're going to separate from one another in meiosis. One is going to end up in each gamete. So here we have, gene A. Here we have gene B. And so in this case, it doesn't really matter which one goes. We're going to end up with a gamete that has a capital A, capital B. Same thing here, homozygous recessive. So we're going to end up with, you can see, either lowercase a, lowercase b, or lowercase a, lowercase b, or lowercase a, lowercase b, or lowercase a, lowercase b. So again, it doesn't really matter. You're going to end up with lowercase a, lowercase b, and you won't have any way to distinguish which alleles ended up in that gamete. However, well, I shouldn't say however. Then what we could see is now we can actually see the law of independent assortment, because now in the F1s we are heterozygous for both the A gene and the B gene. And so what Mendel saw and what we learned as the law of independent assortment is that you can see that now any of these combinations are possible, right? So we could have capital A, capital B right there. And then we could have capital A, lowercase b, that's right there. We could have lowercase a, capital B, that's right there. Or we could have lowercase a, lowercase b, so that's right there. And so these are grouped on the left because they represent the P1 generation gametes as well. So we would call these sort of like parental gametes, because they're in the same vein as the parental gametes, whereas these we call recombinant gametes. And so independent separation of loci of these different genes results in recombination. And so you can see, like I said, new combinations of alleles give us recombinant gametes. And so through recombination, gametes contain new combinations of alleles. This is what Mendel saw because he was dealing with, and there was no way he could have known this, but he was lucky enough to pick 7 different traits, and the genes responsible for those 7 different traits happen to be on different chromosomes. So he never had any problem of these genes kind of interacting with one another. They assorted independently into gametes and therefore independently into the offspring because they were on different chromosomes. So again, this idea of recombination is nothing new, right? We're doing a little review right now. So recombination is the sorting of alleles into new combinations. And so we saw that, right? We came up with every possible combination whenever we set up our Punnett square. Every single square was a possibility. Some of them obviously led to the same phenotype, so that skewed our ratios of phenotypes, but all different combinations were possible. We just assumed that they were possible. We were assuming independent assortment. And so what that means is that the combination of alleles in an organism's gametes are going to be, you can see, they're basically going to be sometimes parental, sometimes recombinant. And so they can be recombinant and with independent assortment, and that means that they can differ. So they may differ from the combination of alleles from its parents. So you have an organism's gametes containing, so the F1 gametes containing the parental same sort of combinations of alleles, and then you also have new recombinant gametes. So now we can restate Mendel's principle in terms, or principles in terms of chromosomes. So segregation is that diploid organisms possess two alleles for a trait, each at the same locus on different homologous chromosomes. I shouldn't say different, right? On two homologous chromosomes. They're chromosome 3 and chromosome 3. So they are homologous. They're not identical, but they are homologous. And so what we saw is that the chromosomes are going to segregate during meiosis. They're going to be separated from one another, and that's what gives you all those different combinations. And so each gamete receives one of the two homologs. Independent assortment is that, in meiosis, what happens is that each pair of homologous chromosomes assort independently into other independently of other homologous pairs. So what that means is that in addition to any combination being possible, then now whatever chromosomes you have, they are going to go to a particular gamete. Let's say you have 4 different gametes that are going to be created, or at least the nuclei for 4. And so gene, the capital A allele on one chromosome goes to gamete 1. That has no bearing whatsoever on where the other gametes are, where the other chromosomes are going to go, which gamete, which combination. They don't care. They're all acting independently of one another. And so now here's the question. What if some genes are on the same chromosome? If the gene A allele, capital A allele of gene A on, what did I say, chromosome 3, if that goes into the, let's say, gamete 1, well, it doesn't matter what's happening with the other chromosomes. But if there's another gene on chromosome 3, it's possible that it's going to go with the A gene on chromosome 3. They're going to travel together. And so here's the thing. In human beings, for example, we have 46 chromosomes, right? We have 23 pairs of chromosomes. We have somewhere between 20 and 25,000 genes. That means that we are going to have more than one gene per chromosome. And so it's impossible. You can't have all the different genes assort independently. Some of them are going to travel together. And so the ones that travel together are called linked genes. Linked genes do not assort independently. Linked genes are not something that Gregor Mendel ran into. Linked genes are genes located close together on the same chromosome. If they're very far apart on the same chromosome, well, that's different. They could get separated in a moment. I'll show you. They could get separated by a recombination event, right? A crossover event. But if they're close together on the same chromosome, they're usually going to travel together in meiosis and end up in the same gametes. And so they are going to, we say, they belong to the same linkage group. And as I've said a few times out loud, here it is in a bullet point. They travel together. So 2 different genes, 2 alleles of 2 different genes, one allele per gene. If they're close together on the same chromosome, they travel together during meiosis, eventually ending up in the same gamete because they're on a chunk of a chromosome that is going to eventually end up in that particular gamete, and so they're going to travel together. Now, this is not expected to assort independently. You don't expect them to assort independently because they're so close together. It's highly unlikely that a random crossover event is going to occur between them, and so they're not going to get separated from one another. They're going to travel together. Miraculously, Mendel examined 7 characteristics that all did assort independently. He didn't know, but they were on different chromosomes. They were not close together on the same chromosomes. And so he had no idea that certain genes could be linked. He didn't run into that. That wasn't his problem. And that's fine. That would have thrown off some ratios. But and mathematically, it would have made it very difficult to figure out to decipher his data. But we now know of linked genes, genes that travel together through meiosis because they are close together on the same chromosome. So many characteristics do not adhere. So like linkage, many characteristics do not adhere strictly to Mendel's second law, the law of independent assortment. So let's take a look at an example from your book. What we have is something that's a little easier to follow because they're kind of visual phenotypes. So we've got some purple flowers so a plant that produces purple flowers and long pollen grains. And then we have a plant that produces red flowers and round pollen. And if we were to cross, so if they're homozygous strains, so this is, well, I guess from the F1 generation, we could tell this is homozygous dominant. So homozygous dominant for purple flowers, homozygous dominant for long pollen, homozygous recessive for red flowers, homozygous recessive for short pollen or round pollen. And so then here we have heterozygous, right? So we're going to have, whatever. So let's go capital P, lowercase p, and long pollen. So capital L, lowercase l, or something to that effect. So the F1s are all showing the dominant phenotype as you would expect in 100% of the F1s. This is still following Mendelian inheritance and what he saw. And so they're all purple, they're all long, and so these traits must be dominant. But then if you go past that F1 generation, you go to the F2 generation, you might recall we used to see a 9-3-3-1 ratio when we looked at, purple versus red and long versus round pollen. And so he did not, or not he, but one does not see 9-3-3-1 ratios in the F2s when you're dealing with purple flowers and long pollen. So what's going on there? Because this was self-fertilization. It's not like we crossed anything. In fact, if you look at it, we see quite a few of a parental phenotype. And the next largest value is going to be of the other parental phenotype. So you have parental, and just a little bit of the recombinant phenotype. So if we do the math, we see a 13.5, 1, 1, 2.6. What's going on there, right? So why is that? And the short answer is, I guess the gene for the flower color and the gene for the shape of the pollen grain are somewhat linked to one another, because they tend to, if you just counted up when they were stuck together, when they remained stuck together and did nothing, then you would have 284 plus 55 versus 42. Right, and so that's going to be quite a bit of skewing towards the parental generations or the parental phenotypes as opposed to the new recombinant phenotype. So we would say that the way to explain this is that these two loci for separate genes must be close to one another on the same chromosome so that they could be in the same linkage group. That's what we mean by linkage group. A linkage group is basically they are linked. If you look at statistically their distribution, you will find that they are not going to distribute according to classical Mendelian ratios because they are linked to one another. And so we say that they are either completely linked, or they're going to be incompletely linked, or they're going to be unlinked. Incompletely linked is the biggest category. It could be anything. Completely linked is when you see them almost always. That would be, if they were completely linked, they would never get separated, and so we would basically see like 0 and 0. But if they're incompletely linked, then we could see any skewing of these numbers such that you're usually still going to see, if there's a little bit of linkage, still going to see greater number of the parental phenotypes in the F2s than the recombinant phenotypes. And so the take home here is that linked genes do not assort independently, which is what it says up here. Linked genes do not assort independently. So if linked genes do not assort independently, then linked genes close to one another are going to usually be inherited together. The only thing that can really separate them is going to be crossing over. Crossing over can prevent this. Crossing over is the antidote to linkage. Not that there's anything wrong with linkage. Crossing over is the way to undo linkage, the way to prevent linkage from occurring. And so my question to you is, when do you think this happens? So you have to promote crossover events to combat gene linkage, or if you want it to be linked, then you'd have genes very close together and crossover events really couldn't do anything. But regardless of all of that, when is it possible for a crossover event to prevent gene linkage? Well, it turns out that we're looking at meiosis 1, right? So late prophase 1 is when all this crossing over is going on, and then they line up at metaphase, and then they get torn apart. So once you get to anaphase 1, then there is no chance of crossing over anymore. You could still maybe get something done in metaphase, but realistically, it's going to be late prophase, early metaphase. And so then when you get to metaphase 2, nothing happens, right? So here there's no crossover events, right? We're not lining up like this to induce a crossover. There's no synapses. So genes occasionally switch from one homologous chromosome to another. So like, for example, these are, genes that are homologous to the genes on the other side because everybody's lined up. And so if you switch a gene from chromosome 19 to the other chromosome 19, that's just homologous recombination. It could be like a dominant allele gets swapped over, and so now you have a dominant allele where you used to have a recessive allele. The other organism gets the recessive allele. Anything like that. And so this results in recombination, homologous recombination. So it breaks up combinations of alleles of genes close together on the same chromosome. The only way you can separate linked genes properly in a biological system is to have them undergo a crossover event where they actually swap parts. So, to summarize, linkage and crossing over basically have opposite effects. Linkage keeps genes together, skews Mendelian ratios. Crossing over mixes genes up, gets us closer and closer to our original 9:3:3:1 or 3:1 ratios, Mendelian ratios. All right, so now that we have a general idea of what crossing over is, or crosses, or just when we're talking about the different alleles of different genes on the same chromosome. When you're dealing with crosses with linkage, it's important to know the genotypes and the arrangement of genes on chromosomes. So we have been writing things without this information. We've written a cross between a homozygous dominant and homozygous recessive as this. But there's problems with this notation. Like, for example, chromosome A, or whatever, the chromosome carrying gene A doesn't have two copies of it. So that means that this must be a chromosome, like let's say chromosome 7. This must be chromosome 7. This is chromosome 7, the other one. This is chromosome 7. This is the other one, chromosome 7. So it would be better to write AB, AB, because that would be more indicative of the chromosomes themselves. So for linked genes, we do that. We write the specific alleles as they are arranged on each homologous chromosome. And so you can see this line represents a chromosome, so AB. Here's another one, the homologous chromosome AB. So homozygous dominant is going to be crossed with homozygous recessive, which is right here, but here we're showing it with linkage. And what you're going to get is you could get the parental outcomes. Right, so this one line is representative of these two, right? These both represent homologous chromosomes. So it gets kind of redundant, so people start using just one line to represent the homologous chromosomes. And you can see that on this chromosome, we have gene A positioned against another copy of gene A dominant, B against a copy of dominant B. And so that's going to be your phenotype, right? Capital A, capital B. Except for when you have a homozygous recessive organism, or at least homozygous recessive genotype. So we can even further simplify this and draw it as AB/AB, but that's really like taking something like this and making it really easy to type, so it really speeds things up, but it loses a lot of information. So you won't see me using this notation very often. I just wanted to mention it because you should know it exists. And then just two more little things to be aware of when talking about linkage. Sometimes you're going to have what's called a cis configuration, or genes are going to be coupled or in coupling arrangement. And what that means is that when you have your wild type alleles that are on one chromosome and mutant alleles on the other chromosome, well, then you're kind of grouping the wild type alleles on one chromosome and the mutant alleles with themselves, and that keeps them away from one another. That keeps the wild type together with the wild type and the mutant with the mutant. Hence, that's why cis means same, right? So you keep the same type of alleles together and the same type of alleles together. Of course, now, if a crossover event were to occur, that would swap some of those alleles, and so then you end up with, say, for example, a wild type gene A and a mutant gene B on the same chromosome. Well, you would no longer be in cis, and so you probably are quite aware that usually when we have cis means same, then that goes with trans. And so the trans configuration is what I just described. It's also called repulsion. In fact, repulsion, you can see how here I put cis first and coupling second, and here I put repulsion first and trans second. Cis is very common, and so you'll hear cis a lot when dealing with linkage, or if you're reading about linkage, or if you look at linkage any further in other courses. Repulsion is the term that you'll see more often when dealing with like, for example, on a previous slide, we had a capital A, lowercase b. Let's call capital A the wild type version of that gene, the wild type allele of that gene, and the lowercase b the mutant allele of the B gene. That would be an example of repulsion. So repulsion, or the trans configuration, it occurs when a wild type allele and a mutant allele of a different gene are found on the same chromosome. So capital A, lowercase b, capital well, let's try lowercase a, capital B. Both of those would be examples of trans configuration or repulsion. All right, now that we've got the terminology out of the way, let's start with an instance of complete linkage. Now, it's important that you realize that we're going to talk about complete linkage and incomplete linkage. Incomplete linkage is going to be something that we're going to see a little bit more often when dealing with linked genes than complete linkage. Now, if we have unlinked genes, then that's totally different. That's what we've been talking about thus far, actually, in the course. We've been talking about alleles of genes, different genes that we're assorting independently, so those show no linkage. But let's start with the less prevalent complete linkage, because honestly, it's a great way to show what linkage is. So complete linkage is the phenomenon in which different genes are located very close to one another on the same chromosome. So they are very, very rarely going to be separated from one another. They are going to stick together most of the time. And the main reason why this is an easy statement to make is if you understand that if the crossover events that could separate these two genes occur at random all up and down the chromosome, the closer genes are to one another, the less target area, the less likelihood there is that a random crossover event will occur between them to separate them. Think of it as lightning striking, right? So if you said, well, how often does lightning strike in the state of California? Well, then you have a certain, that's, you can measure that, you can figure it out, and you would expect it to strike, fairly decently. I understand, the weather patterns and all that, but that's going to be a far greater, the number of lightning strikes that you'll measure throughout the state of California is going to be far greater than if you said in San Luis Obispo County. And then if you even further specified and you said, okay, well, how often does lightning strike at, 1 Grand Avenue, right? Like you just pick one place, one address, or then you can get really crazy and you can like put a little, like, I don't know, take a quarter and you put that down on the ground and you say, how often does lightning strike right here, right? And so the less area that you provide, the less likelihood of a lightning strike occurring there, because lightning is striking somewhat randomly, right? At least it's random in my analogy. And so same thing on a chromosome. So these crossover events occur randomly all up and down the chromosome. And there's a lot of them, but they occur randomly. So if you have genes that are on completely opposite ends of the chromosome, so if we had, we take one person and we stick them way up north, right? Northern California, up like past Eureka, way up north. And then we've got another person down in Chula Vista, down, way down south. And you say, how often is lightning going to strike between these two people every year, right? It's going to strike a lot. And so if each lightning strike is a possibility of separating them, my analogy's falling apart at this point, but it's the best I could do without a board and having you in front of me in class. But basically, you get the idea, right? So if there's a tremendous amount of space between them, then there's a high likelihood of lightning striking between them. And if lightning strikes between them, that's like a crossover event, and that crossover event will separate them. They could travel on different chromosomes. Whereas if they are standing face to face, anywhere, you pick a place. I don't know. Let's just go with like Bakersfield. That's kind of middle-ish. And so if they're standing face to face, so actually, no, it's not. I guess it would have to go a little further north. What do you think? Fresno? Fresno sounds good. Let's go with Fresno. So they're standing face to face in Fresno. And we say, how often is lightning going to strike between them? I mean, it's almost never going to strike between them, right? Well, then in that scenario, they would be completely linked. So I'll say it one more time. Complete linkage is the phenomenon in which different genes are located very close to one another, face to face in Fresno, on the same chromosome. And so because they're very close to one another, they do not exhibit crossing over. The crossing over events occur at random, and if they're very close to one another, that random crossover event is unlikely to occur. It's very unlikely to occur between completely linked genes. So let's consider a pair of linked genes in tomato plants and see how this plays out. So I grow a lot of plants. Have a pretty big garden, and I'm like an amateur farmer, I guess. And basically, one thing that I actually had this conversation with my children is we were like, yeah, I think we were at Farm Supply or back in the day, Osh, and we were looking at tomato plants because we were just going to put some tomato plants in our garden. They were already grown. Why start with seeds when you could buy plants? And so there were these little tomato plants that didn't look very healthy, but the soil was nice and wet, and I mean their roots were good. I mean they were basically perfectly healthy, and so they actually had mottled leaves. And so you can have mottled leaves. Tomato plants can have mottled leaves, and they look like, if you've ever had a tomato plant that you didn't water and it kind of start to shrivel up, it looks like that, but then if you touch it, you'll feel that it's still soft. It's not crispy. And so that would be a plant with a mottled leaf type as opposed to just one that's drying out and dying. And so we have mottled leaves, which are recessive to the normal leaves that everybody's going to go for. And so I remember specifically what I'm talking about is there was one little plant that was like kind of left alone because everybody was picking out the ones that were taller and had the more vibrant and I don't know how to describe it, more normal leaves, so like smoother leaves. And and so one of my little girls said, "Oh, well, we don't want that one. It doesn't look very healthy." And I'm like, "Actually," and then I went into my whole mottled leaf spiel, and for her, in her defense, she did listen for as long as, a 5-year-old is going to listen. And so anyway, and I ended up buying the plant because it had a whole bunch of little flowers on it, so it was going to make a lot of tomatoes, and it did fine. It did great. So that's besides the point. That's just a phenotype, right? So mottled leaves are recessive to normal leaves. I'll show you pictures here in a second. And plant height, right? So dwarf is recessive to tall. So normal tall is going to be apparently a doctor, right? It's going to be MD. And mottled dwarf is going to be lowercase m, lowercase d. And so if we were to test, right? So let's think about the genetics of this. Let's throw in a little bit of the genetics that we've already learned. If we were testing for linkage, we could do that with a test cross, right? We're going to cross these two plants. And if we're testing for linkage, we're going to need a specific type of plant, right? We don't want to cross like a homozygous dominant with a homozygous recessive, and then try and like determine linkage, because what we would expect is the phenotype's going to be all dominant, right? And so then we could use that to test a little bit for linkage, but it's going to be a little tougher. So what we really need to test for linkage is one of these two things, right? So what we need is a plant that is going to be heterozygous for both characteristics. What we want is a big M, big D, little d plant. And if you're writing this, down, write down big M, big D, little d, and we're going to cross that with all homozygous recessive. So little m, little d, little e. And the reason why we're going to do that, usually I do this on the board and I ask questions of the class at this point, but I'll just tell you, the reason why we're going to do that is because we can follow where the one big M allele and the one big D allele goes, because they are going to contribute to an observable phenotype. And they're the only ones that are going to contribute a normal leaf type and a tall plant. And so we can check to see if normal tall gets separated from one another. And we have like a normal leaf plant that ends up being dwarf, or a mottled leaf plant that ends up being tall, right? So we could see if they actually get separated from one another. So we would obtain this heterozygote by crossing homozygous dominant plant with homozygous recessive plant. It's going to start off the same as everything we've done before, and I'll show you that on the next slide, but this time we'll use the linkage nomenclature. We won't say big M, big D, little d. We'll say, oh, okay, well, on this chromosome, we have, let's say we're doing it in cis, right? So we have big M, big D. And on the other chromosome, big M, big D. So that's our homozygous dominant parent. And then little m, little d, little m, little d on our homozygous recessive parent. That's how this cross is going to look. So let's take a look. Okay, so this is, like I said, this is what we used to write, right? This is the way we used to write it. This is not using the linkage nomenclature. I'll show you that in just a second. So this would be our homozygous dominant parent for the M gene and the D gene. So this means normal leaves and tall. This means mottled and dwarf. And so this would be the P1 cross, and then for the F1 cross, we would expect the heterozygote that we're looking for. And you can see that here, right? In the F1s, all of the F1s are going to end up looking like this. But let's do this with the linkage designations, writing it out as if we're actually trying to depict linkage. So exact same cross, but now we are depicting it using what we just learned about how to show, how to show linkage. So what we have is the exact same thing we had on the previous slide. So we've got our homozygous dominant parent. So you can see they're diploid. They have one. Remember, this line represents a chromosome. So above this one chromosome has big M, big D, and then this line can be used to represent the other chromosome, which is big M, big D. So they're homozygous dominant, homozygous recessive, and then the F1 is going to have basically big M, big D, little m, little d. So it's going to have a chromosome with big M, big D, chromosome with little m, little d, because we're thinking complete linkage, right? I'm showing you an example of complete linkage. And so now, if we want to test this idea of complete linkage, we can use this F1 heterozygote and we can perform a test cross. You could actually even call this a back cross. This test cross is going to be basically cross the F1 and test for linkage. See if the normal tall gets separated from model dwarf. Like if you end up with normal dwarf and model tall or something like that. So we can use the F1 heterozygote to perform a test cross, test cross for linkage by crossing it with a homozygous recessive. So that's crossing it with this genotype, which is why you could even call this a back cross. It is a test cross because you're testing something, but you could also call it a back cross because you're crossing back against one of the parental genotypes. And so here's what that cross looks like. So we took the F1, we're crossing it with an all homozygous recessive, and the whole point is what we're starting with is perhaps what seems to be normal, normal tall. And we want to know whether the normal and tall phenotypes can be separated from one another. Whether, as I've said a few times before, whether we could have a normal dwarf plant or a modeled tall plant, or if they're all just going to end up normal tall or modeled dwarf. So we do the cross. And if this is the outcome, then we would say, oh, no, looks like normal, and so I guess normal and tall stayed together. They stuck together. So let's use some numbers to try and make sense of this. So note that the heterozygote produces two types of gametes. That's why we care about the heterozygote. It could make, theoretically, this gamete or this gamete. And so because there is no crossing over because of complete linkage, if there was complete linkage, we're testing for it, but I'm just telling you this is a complete linkage example. So we're testing for it, and if we do this cross and we see, you could see the outcomes given, right? So if we see that we get a normal tall plant and we get a model dwarf plant, and that's it, those are the only kind of plants we get, we see no crossing over. We don't see the recombinant phenotypes like a normal dwarf or modeled tall. And so these are the only gametes produced by the heterozygote. So no, as I said before, let's see, normal dwarf and modeled tall. That didn't happen. So like I said, we need some numbers. So here is, I mean, they're not numbers per se, but they are at least a visual. And so here's what we started with. We started with normal tall. And we started with a perfectly good tomato plant, by the way. Just because it has mottled leaves and it's dwarf doesn't mean it's not going to make potatoes. Well, it's not going to make potatoes. Doesn't mean it's not going to make tomatoes. And so what we've got is normal tall crossed with mottled dwarf. That gives us, upon fertilization, we do this cross. So this is the heterozygote, right? So this is not the homozygous dominant. We're not looking at the P1 generation. We're looking at the cross that we just did, the test cross. And so what we've got is our heterozygote, and we have our homozygous recessive. And so we do that cross, and then we see what we get. And if we get all non-recombinant progeny, if we get all normal tall and mottled dwarf, well, then that means that we get a 50-50 distribution. And so that is a perfect example of complete linkage. Only non-recombinant progeny are produced. If you have no recombinant progeny, then you have complete linkage. Now, if this was, happening according to what we've learned before, where you had independent assortment, we would expect to see 4 different phenotypes. You would see that here, right? We would expect to see a quarter of them would be non-recombinant from this specific cross, right? So this is not exactly like we used to do heterozygote, heterozygote, but now we're doing heterozygote, homozygous dominant. Oh, let me try that again. Homozygous recessive. So heterozygote, homozygous recessive. And so if you work that cross out on your Punnett square, then you would expect to get a quarter that are normal tall, a quarter that are mottled dwarf, a quarter that are normal dwarf, and a quarter that are mottled tall. Make sure I got that right. Oh, it says it right here. I should have just read that. Anyway, that would be, this would tell you, oh, okay, well then if you have a quarter, that means you have 25, you have an equal percentage chance of any one of these. That would be a conclusion that you would get, which would be independent assortments happening here. And so half the progeny are recombinant, half the progeny are not recombinant. So there's no skewing towards non-recombinant or recombinant. And so basically what you have is independent assortment, any possibility. But that's not what we see. What we see is exactly the same thing. Normal sticks to tall, mottled sticks to dwarf. Those two phenotypes stick together. That tells us that we're looking at complete linkage. So there are they are non-recombinant gametes because you're getting non-recombinant progeny. Sometimes non-recombinant gametes are also called parental gametes, but that kind of throws some people off. They think that they're gametes from the parental, like the P1 generation. What there really are gametes that mimic the P1 generation, that are the same as the P1 generation. So I prefer non-recombinant gametes, but you will run into parental gametes in the book on, different questions, things like that. So the progeny all display the original combination of traits and the original combination of traits that you saw in the parents. So therefore, they are non-recombinant progeny. Therefore, we conclude from this example that the 2 genes are linked. They are completely linked because we're not seeing any recombinant progeny. If we saw a few, we would say, well, they're not 100% linked. There's a teeny, maybe, they're very, very close to completely linked, but not so much. And so they would be incompletely linked. There are many different degrees of incompletely linked genes. So let's look at incomplete linkage. So the way you get from complete linkage to incomplete linkage is by crossing over. There has to be crossover events. Those crossover events can be maximized if you have more and more and more DNA sequence between your genes of interest on a particular chromosome. So usually there is some crossing over between genes on the same chromosome producing new combinations because they're not literally adjacent to one another. There's some distance, there's some amount of sequence between them. They could be close, but they're not right on top of one another. And so genes that exhibit crossing over in any fashion, any amount, would be incompletely linked. If they exhibit it all the time, then they would be completely unlinked genes. We wouldn't be talking about them in terms of complete linkage or linked genes. They're just completely different, what are called linkage groups, meaning they could be on different chromosomes for all that we know. But right now we're talking about genes that exhibit crossing over, but the Mendelian ratios are not maintained. You'll still see more non-recombinant outcomes than recombinant outcomes, not an equal chance of non-recombinant or recombinant, like we saw with that hypothetical with the tomato plants, right? Quarter, 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 quarter. Hopefully I said that 4 times, right? Quarter, 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 4 of them, yeah. So, so basically, if that's the case, then you have incompletely linked genes. So crossing over, this is very important. We talked about this before. The reason why I went over the cell cycle early on in the course is because it is never going to go away. It is important in many aspects of many topics that we'll be talking about throughout the rest of this course. And here it is again, right? So we're back to meiosis, and we're talking about crossing over. Remember, crossing over is the exchange of genetic material between non-sister chromatids during prophase 1 of meiosis. These are, okay, so this red one is a sister chromatid of this red one. This blue chromatid is a sister chromatid of this blue one, but red and blue are the non-sister chromatids that we're talking about. And one thing you need to remember is remember the metaphase plate. Remember that whenever it could be metaphase, 1, metaphase 2, I don't care, or just metaphase of mitosis. But here we're talking about meiosis. So metaphase 1, metaphase 2. In this case, actually, it would be metaphase 1 because we need the cell to still be diploid. Remember, meiosis 2 is going to be, they get separated from one another, right? So don't get thrown off by this image. It's not saying that this is happening in meiosis 2. This is, okay, so in metaphase, so late prophase 1, early metaphase 1, you have, on the metaphase plate, it's a plate. When you think of a plate, think of a two-dimensional object, a plate that you set things down on, right? And so that's what I mean by two-dimensional. I know a plate, you can lift it up, it's 3 dimensions, I got you. But basically, when you, like here, the best way to do this is to just take your fingers, and you're using your index finger and your middle finger, right? And so your left hand, your right hand, have your palms facing away from you. You can, like make a little, like almost like a peace sign with each hand, and like you're telling somebody, obviously this would be better if we were in person, but you're making a peace sign, right? So you're making a peace sign. So put your two fingers together, that making the peace sign. So now you just have, I don't know, you're calling for somebody, I don't know. But the point is, now take your left hand and your right hand and then put them together. And if you do that, to simulate like a metaphase plate, what you should have is you have basically your ring finger and pinky finger being held down by your thumbs, and then your palms are facing, so like this, you're looking at the back of your hands, you've got your middle fingers and your index fingers up, and they're up against each other, and what you'll notice is only your index fingers are touching one another, right? So you're not trying to put them against each other. You can't, face them one another, because your thumb and your pinky finger and your ring finger, those things get in the way, so they can't come in contact. So you have to touch your index fingers together. So now you're looking at the back of your two hands touching one another, and hopefully you're playing along, because if you're not playing along, then this is just craziness. But you're looking at the back of your hands with your two fingers from each hand pointing straight up, and then it's at metaphase, so they're stuck together, or prophase for that matter, prophase 1, I should say, prophase 1. And so now your index fingers are touching, right? Your middle fingers they're not going to touch each other. And so you can try and make them touch each other, but it realistically in a cell, the chromatids are not going to touch each other, the outside chromatids. So what we're talking about is this would be a middle finger, this would be a middle finger, this would be an index finger, this would be an index finger. And so because your index fingers are in contact with one another, or non-sister chromatids are in contact with one another, they can exchange parts. Crossing over can occur there, and you can see that here. So no crossing over, but here you can see these two chromatids can exchange parts. And they're just showing one crossover event. Remember, there's just countless crossover events occurring all up and down these chromatids. But these two can cross over, right? So the red and the blue here can cross over. This outside chromatid and this outside chromatid do not have access to one another, so there's not any crossover event there. This is why it was so important that I was trying to emphasize, make sure you're comfortable with picturing what's happening during meiosis and the metaphase 1, prophase 1, metaphase 1, or homologous recombination when they pair up in synapse, because that's going to play into the numbers now. So here's my question to you. If these two chromatids are the only ones that are capable of undergoing a crossover event, what is the maximum percentage of recombinant chromatids you can get? And so hopefully you can just look right here and you can see that for any homologous pair of chromosomes, the maximum number of recombinant chromatids you can get, because the outer ones don't have access to each other, is 2 out of 4, so 50%. If a recombination event occurred every single time to separate A and B genes, alleles of these genes, right, to basically recombine them so that you have different combinations, then the maximum amount of recombination you would measure is 50%, because the outside chromatids are not going to be involved in crossover events. Now that 50% is very important for recombination frequency. So we're going to learn about recombination frequency and how to map this. And recombination frequency, as you'll see in a moment, directly relates back to map distances on a recombination map. So we can map, or Thomas Hunt Morgan and his students did a while back, figure out a way to figure out where genes were relative to one another. So, it's been decades since I took the SAT test. Some of you probably have taken it much more recently. I don't know if they still have questions like this, but I distinctly remember questions that I was like, oh, I got these, right? There were certain questions that I was like happy to see. And there were like ranking people in order of their height, right? Those kind of questions, and maybe not the SAT, but maybe you've seen these before where it's like, I'll just use the generic names that I remember from when I took it, right? So like Tom is taller than Sally. Sally is shorter than Phil. Phil is taller than Julia. Julia is taller than Tom, or something like that, right? And I know I just made that up, so it probably doesn't work. But then you had to rank them in order of their height, right? That's kind of like what we're talking about here. We're going to talk about basically what are called genetic maps. And genetic maps are going to be basically maps based off of recombination frequency. And so they are going to have a maximum recombination frequency between two genes of 50%. If it's 50%, then they're always separated. And every possible time they could be separated, they get separated because 50% is the most you can separate. And so then basically at that point, we don't know if they're on the same chromosome or on different chromosomes because you'd get the same numbers. So 50% means no linkage whatsoever. Anything less than, say, 45, because, our math isn't great. I'll talk about that in a moment. But anything less than 45%, you're looking at incomplete linkage. And if you get down to 0%, well, then, okay, that's complete linkage. So for closely linked genes, crossing over does not take place every meiosis. They don't get separated, and so you're looking at, you're going to end up, so you can imagine, if you had no, if you had no crossing over, so it's gone now, but if you have no crossing over, then you'd end up with basically non-recombinant, 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 non-recombinant. But here we have a little bit of crossing over. So for closely linked genes, crossing over does not take place every meiosis. But even if it took place every single time, like in this case, every single time if A and B got separated from one another, then we still only net out 50% of the resulting gametes would be recombinant, right? This chromosome would end up in a gamete non-recombinant. This chromosome would end up in a gamete recombinant. This chromosome would end up in a gamete recombinant. This chromosome would end up in a gamete non-recombinant, so 50%. So hopefully, it's clearer to you now that you're going to see this number, right? You're going to see recombination frequencies, and they're never supposed to be higher than 50%. If it's higher than 50%, you did something wrong. Just like, if you're working really hard and I can think of an example. So if you've ever worked in a sort of laboratory setting and you had to take your yield, right? You had to determine your yield. If you come out with a 306% yield, that means that something's wrong, right? Because you're not going to end up with more than is physically possible that you started with, right? 100% yield would be phenomenal, and you don't expect to get 100% yield, but 306 means, okay, either I did something wrong or my calculation's wrong, right? Maybe I have extra water weight that is throwing off my calculation. In the case of linked genes and crossing over, 50% is the maximum recombination frequency. Okay, so how do we calculate recombination frequency? How do you know what percentage you have? Recombination frequency is actually pretty easy to calculate. It's the percentage of recombinant progeny produced in a cross. So we saw previously 2 out of 4, 50%. So recombinant frequency is basically going to be the number of recombinant progeny divided by the total number of progeny, total number of progeny, times 100%, of course. And so recombination frequency was used by Thomas Hunt Morgan and his students to devise the first genetic maps of chromosomes. So this was a long time ago. This was 1900s, early 1900s. And he had a lab. It's a funny story. I'll keep it really short. He had a lab in Columbia, or at Columbia University. And he was a young, no name, not as prestigious named professor yet. And so he worked with Drosophila. He worked with fruit flies. And I think I may have mentioned this previously. Fruit flies, if you work with fruit flies, they get out, right? And so if you work on a floor in a building, or anywhere actually in a building with fruit flies, some other lab works with fruit flies, those little bastards are going to find their way into your lab, and then while you're working or trying to write, they're just going to sit there buzzing your face, right? They're just like interested in annoying the hell out of you. And so Thomas Hunt Morgan's lab was a Drosophila lab, a fruit fly lab, and he hadn't really, become the big name that he is now. I mean, like, map units are named after him now. They're called Morgans and centimorgans. But basically, this was before that. So they kind of banished him. Like his peers were like, okay, the fruit flies are out of hand. You need to get out of here. And so they like banished him to this older building, and he was way up high, and it was drafty, and so if the fruit flies got out, it was no big deal because they would just leave. They'd just go out into the environment as opposed to staying in the building. And I'm just picturing like this old, like clock tower kind of setting. But then he and his students devised a way to figure out where genes were relative to one another. And then he became Big Bad Thomas Hunt Morgan. So what he and his students did, and I always say his students because it's really sad, actually. You never hear about what some of their names are. You just hear about his graduate students, and for good reason. I mean, it's his laboratory. He, was providing all the guidance and everything. But some of them had some really good insights, and it'd be nice if we knew their names, right? So, but anyway, so Morgan and his students, his graduate students, developed the idea that physical distances, so the actual distance between genes on a chromosome, are related to the rates of recombination. Now, I've been saying this the entire time, and so obviously, I didn't come up with this, but here's the idea. Recombination events occur randomly up and down the chromosome. The farther apart genes are, the more likelihood that a recombination event will occur between them, and that could separate them. That could make them travel not together. They could travel independently in meiosis and therefore end up in different gametes. The closer they are together, right, once they get face-to-face in Fresno, then a recombination event is very unlikely to occur between them because you're reducing the probability of a recombination event occurring between them as they get closer and closer. And so if that's the case, then that means that recombination frequency is a direct measure of the distance between genes. So that's what this relationship is. So I should have, shown this bullet point before I answered that question. What is this relationship? It's this. It's recombination frequency is basically the physical distance between genes on the same chromosome. Or if they're on different chromosomes, we wouldn't know because the recombination frequency would just be 50%. So they hypothesized, Thomas Hunt Morgan and his students hypothesized crossover events, or crossover event. Each crossover event occurred more or less at random up and down the chromosome. In fact, it's fairly random. There are certain areas of DNA sequence in our chromosomes that are more susceptible to crossover events. But basically, they assumed that they occurred at random, and in fact, mostly they do. And so then they were able to measure recombination frequencies between, different genes. So like, for example, the gene responsible for a yellow body and white eyes, right? So they found that those kind of went together a lot, right? Because they only ended up with a recombination frequency of 10%. Whereas you could see now, okay, remember numbers, 50% should be the highest, but of course, remember that this is experimental data, so you could be a little off, right? Some of you, I'm sure, have gotten more than 100% yield in lab before by accident. And so what you can see here is that you can actually see where genes are relative to one another based off of their recombination frequency. So the idea here is We don't know what the DNA sequence is in between them, so how many base pairs between them, but we get a rough idea of how close or far away they are from one another. Like if I told you that there are, I'm just going to make up numbers here. So if there are, let's say 500 lightning strikes between Eureka and or Crescent City and Chula Vista in California per year, but there are only 3 lightning strikes between San Luis Obispo and Paso Robles per year. Given that you know that lightning strikes at random, you would think, oh, well, then there are probably these cities. Hopefully, at least in my analogy, this relates back to recombination frequency, in that you would think, oh, well, San Luis and Paso Robles must be closer together because there are fewer lightning strikes. So if I said, there are a finite number of lightning strikes, there are 500 lightning strikes in the state of California for a particular year. And between, what did I say, Crescent City and Chula Vista, there were 500. Well, that makes sense because we pretty much covered the entire state. But then if I said there were just like 2 between Paso Robles and San Luis, well, that means, oh, well, we're not covering the entire state, because what happened to the other 498 happened elsewhere. So 2 genes far apart on the same chromosome are more likely to undergo a crossover event. Genes closer together, meaning linked genes, are less on the same chromosome, are less likely to undergo a crossover event. And so Thomas Hunt Morgan and his students proposed recombination frequencies would give estimates of the relative distance between genes. And so, as I said a moment ago, chromosome maps calculated by using recombination frequencies are genetic maps. Now, this is an extremely important thing to make sure that you understand. So, let me say it a few times so that we can be absolutely clear. A genetic map is based off of recombination frequency. A physical map is, as this says, maps using exact base pairs between loci. They came much later. Let me say it that way. They came much later with newer technology. It's a completely different map. Okay? It's not the same. And so if you run into a question that compares the two, you want to choose the answer that says that they are 2 different maps that measure different things. So physical maps actually measure the base pairs, the number of base pairs between genes, right? Whereas genetic maps just give you relative, like, recombination frequencies. They measure recombination frequencies. So there's a difference between a map that you can use that's quantitative, a physical map that tells you that there are, and forgive me if I'm wrong, but there are, 35 miles between the San Luis Obispo city limit and the Paso Robles city limit, as opposed to saying Paso Robles is north of San Luis Obispo, and that's it. It doesn't tell you how far. It doesn't tell you, well, I guess how far is pretty much it. So it's a very different way of looking at these maps. So they're both technically kind of like maps. One tells you the exact distance. It gives you like everything in between. The other one's like, yeah, it's that way, right? And so genetic maps are very, very different than physical maps. They are not the same thing. They do not measure the same thing. Genetic maps are really just ways of visualizing recombination frequencies between genes. Physical maps are the actual location of genes on chromosomes according to their DNA sequence. Okay, so now let's go back to genetic maps. Let's go back to gene mapping with recombination frequencies. So Thomas Hunt Morgan and his students basically determined that distances on genetic maps are measured in increments of 1% recombination frequency. And so they called that a centimorgan. So just like, you could call them map units, or you could give Morgan his due and call them centimorgans. Just like in chemistry, you've got molecules, right? And so individual atoms, their masses are the atomic mass, or you could call them Daltons, right? Because John Dalton came up with the idea of the atomic mass unit. And so you could say 1 AMU, or you could say, 1 Dalton, same thing. So here you could say 1 map unit, or you could say 1 centimorgan. Okay, now here's the good news. If you think that this is a lot, you're going to love genetic maps because they are very easy to come up with. They're just little puzzles and they're not that hard. So my students absolutely, generally in class, my students are like, whoa, what are we going to do with all this linkage? This sounds like a big deal, measuring recombination frequencies. And then when we get towards the end, and again, I'll give you I'll do two examples with you on how to construct a genetic map from recombination frequency data. It's smooth sailing after that. It's really easy. So because genetic distances measured with recombination rates are approximately additive, something to keep in mind, remember we are doing this with experimental data, they are approximately additive. So what that means is that we can construct a genetic map from recombination frequencies. When we do construct our genetic map, don't expect everything to add up perfectly. The larger the distance between genes, meaning the greater the recombination frequency, the less accurate it is. And if you're wondering why that is, well, I'll keep it really simple for you. Remember that the way that homologous recombination works is that you're riding along your particular chromatid, and then you're going to basically cross over a bridge to the other chromatid, right? That's what a crossover event does. It basically swaps parts of chromatids. Well, if there's a large distance between genes, and these recombination events occur randomly all up and down the chromosome, well, then you might actually have another event where you cross back to your original chromatid. And so then we had any even number of crossover events between a particular set of genes is going to be misleading because we're going to think that then nothing happened. They didn't get separated. They ended up together. But actually, they got separated, then they got put back, then they got separated, then they got put back, right? And so then, there were multiple crossover events in my little scenario there, and we wouldn't be able to measure that. So just remember the numbers aren't perfect because of multiple crossover events can throw you off. So let's construct a genetic map. All right, so here's the data that we have. So we have recombination frequencies, good, no number over 50%, that's good. We want to construct a genetic map given the following information. Assume all genes are located on the same chromosome. Oh, that's nice. That's a nice piece of given information for us. So they're all on the same chromosome. So we really just have to come up with one line, and then we have to put A, B, C, D on that line with a proper distance between them. So where do you want to start? So basically a whole bunch of tests were done. So test crosses were done between, a test cross to check for linkage between A and B, A and C, A and D, B and C, B and D, and C and D. So of all of these, we have recombination frequencies. Where do you want to start with your making of a genetic map? You want to start with the biggest number. It's always easier to work with the biggest number and then work your way down. So C and D, D and C, doesn't matter which way you put it, are 23 map units apart, 23 centimorgans apart. That's because the recombination frequency is 23%. So let's start there. Let's draw a long line, and then we'll put D and C or C and D on the ends. This is a distance of 23 map units, okay, approximately. All right. And it doesn't matter if you do C and D, D and C, it doesn't matter. They're basically... the next letter will matter, but C and D are basically our bookends. They're the ones that are farthest apart, so that's what we're working with. At least from what I can tell, there are bookends. We'll see. We might have to amend this. Okay, so now let's look at the next largest number. So 15. A and C are 15 away. So that means that A could be 15 map units in this direction. But the problem with that is that then A and D would be 23 plus 15, 38. And then we see A and D are 8. So that's not the way we go. We're not going that way. So that tells us that A is going to be somewhere over here, right? It's going to be 15 away from C and 8. So actually, I'm sorry, it's going to be way over here. It's going to be 15 away from C and 8 away from D. So we would put, you just roughly kind of try and make it look, somewhat equitable. So there we go. Now we have A. And so A is, so this is supposed to be 15 away from C and 8 away from D. Okay, now where's B? So A and B are 5 away. So that's good. That tells us that B is either here or here, somewhere, 5 away from A. A and C are 15 away. Okay. Oh, we did A and C. I'm sorry, we're doing B. B and C. B and C are 10 away. That tells us something. So if B and C are 10 away, then B is somewhere in here. B is not on this side of A. And then, B and D are 13 away. Okay, so that makes sense because if it's 10 away from C and 13 away from D, well, D and C add up to 23, 10 and 13 add up to 23, B is somewhere in this vicinity. And so here's our 8 centimorgans. There's B. Okay, so there's B. And so we can fill in our numbers, right? So A and B are 5. B and C are 10. So there's 10. There's 5. Let's add them up. So we have 8. We have 5. That's 13 plus 10. That's 23. 23 is not that high a number, so it's actually fairly accurate. The higher it gets, the less accurate it gets. And so all our numbers work out. That's it. That's how we construct a genetic map. We have the luxury of knowing that they're all in the same chromosome, so we're just working with one line. If we had some numbers up here that were large, then we'd be thinking, oh, maybe we are looking at more than one chromosome. And so that would be like a problem where you'd have multiple lines representing the chromosomes, and you'd have to put the places that you would find the genes. And so that's how you construct a genetic map from the recombination frequency data. Let's do another one. Oh, I'm sorry. Before I do that, let me just mention one thing. So 2 points have to be kept in mind when constructing genetic maps, right? And I've said these aloud, but here we go. Number one, for large frequencies like 50%, you cannot distinguish between genes on different chromosomes or genes located far apart on the same chromosome. If it was 50% between D and C, we wouldn't put them on the same line. They would be in what we call different linkage groups. And so we would make a line for D and a line for C, because if they're 50%, we cannot assume that they're on the same chromosome. We cannot distinguish that unless we start looking at all the other data. Number two, a test cross for two genes far apart on the same chromosome, as I said, tends to underestimate the true physical distance between them. As the distances get larger and larger between these genes, like D and C, it becomes less accurate. And you underestimate because you don't count all of them properly because of double crossover events or even numbered crossover events that throw you off. And so that would be why. So you have even number of crossover events that basically when crossing over is occurring, you don't recognize it, you don't register it because you end up with a non-recombinant progeny. And so you basically count that towards the non-recombinants, even though it is recombinant. And so it has a double recombination event in there somewhere, or an even number of some sort. It could be like, it has 60 of them. That's still going to be an even number. And so your genes are going to basically appear as though they're not separated when they were separated and put back together 30 times. So let's see. Okay, so here's an example of what I'm talking about with a double crossover event, right? So when you have a test cross and your genes are pretty far apart, then you could have a crossover event, and then you could have another crossover event that kind of puts it back. So like you can see, these look like, according to A and B, these look like non-recombinant outcomes, but in fact they are recombinant. It's just we wouldn't register them when we're counting up our progeny. When we're classifying our progeny, we're going to say, oh, this looks like we had, zero recombination because the parental, genotypes or basically gave rise to the parental phenotypes. Those all just stayed the same, right? Producing only non-recombinant genotypes in the gametes. All those parts of the chromosomes have been recombined. We wouldn't notice it. So what could be done to refine such a genetic map? If you wanted to make sure that you actually had numbers that were accurate, what could you do to make sure that your A and B are either not separating or you have double crossover events? Well, the answer is you would just do more crosses, right? You would do more crosses, more test crosses. You would look for a gene, for example, in between there to get some smaller map unit distances so that when you build your genetic map, you've got a bunch of different data points, not just one, a mile away from another, and then hoping that your number in between is accurate. So that's what this says. Test cross for additional genes. Do a 3-point test cross. Now, you're not responsible for the 3-point test cross material in the textbook. And so if you ever have any question, make sure you go to the objectives that I posted on the course website. And it says what the reading is. It says what suggested problems to do in the back of each chapter. And it will also tell you in this case that you are not going to be responsible for the 3-point test cross reading material. So you can stop after a 2-point test cross. Okay, so here's that second example I was promising. So we want to construct a genetic map given the following information from a 2-point test cross. Now that's all it says. So I'm telling you, do not make any assumptions. What that means is do not assume just one chromosome, especially when you see 50, 50, 50. I mean, you've got a bunch of 50s in there, and 50 should be, that should make you have pause. That should basically say, oh, okay, that means that A and B, I have no idea if they are linked or not linked. They're probably not linked. They're going to be in different linkage groups. That's what we're going to say. And so A and C is going to be in different linkage groups. A and D are going to be different linkage groups. B and C are going to be, oh, okay, so those are showing some incomplete linkage. So B and C are together. So that tells me that B and C are in the same linkage group. B and D are in the same linkage group. C and D are in the same. Okay, so B, C, and D are in the same linkage group. A is all by itself. That's what I can just gain from looking at these numbers. So let's put A all by itself. That makes it easy. Now let's look at B and C and D. So we don't care about A. A's done. A's 50 away from B, 50 away from C, 50 away from D. So it's on its own. You know, you can put anywhere on this line. I just put it there. And so you basically could go anywhere on this chromosome because this is, you know, maybe this is a chromosome. Maybe this is just a far away part of the same chromosome. We're just going to call it linkage group 1. Then we have to come up with linkage group 2. Linkage group 2 is going to have B, C, and D in it. So start with your farthest apart, C and D, just like we did before. They're 28 apart. B and D are going to be only 10 apart. B and C are 20 apart. Okay, well, that makes it easy. So that means that B is closer to D than C, so we just build that. And so, I mean, really, we're almost done here. We've got, actually, we are done. This is the linkage group. It's got B, C, and D on it. And B and C are 20 apart, just like they're supposed to be. B and D are 10 apart, just like they're supposed to be. And C and D, D and C, or C and D are 10 plus 20 is 30 apart. But our number here says 28. That's okay, because that's what we've been talking about this entire time. They're pretty far apart. So we would underestimate, remember the previous slide, we would underestimate the recombination frequency. If we did another cross, so we did a, another gene in there, so now we know that actually, so this is 10, this is 20. Those are fairly accurate numbers. 10 especially is much more accurate, right? And so that means that we know that C and D are probably 30% or 30 centimorgans away from one another based off their recombination frequency. So that's how you figure these out. So what we were talking about here is you notice this 28, that's okay because 10 plus 20 makes 30. And so this is just underestimating that a little. That's perfectly acceptable. That's what you would expect because of those double crossover points or double crossover events. Okay, so quick little summary. I think we already, talked about all these things, but let me just say it one last time. 2 points have to be kept in mind when constructing genetic maps from recombination frequencies. Number one, for large frequencies, 50%. I think I already told you we can't distinguish between genes on different chromosomes, and so those are going to be in different linkage groups. For number two, when a test cross for two genes are far apart on the same chromosome, like say for example 28 centimorgans away from one another, you tend to underestimate the true physical distance between them, meaning so they actually turned out to be 30. And so here is just a graphical representation of that, the actual map distance versus the recombination frequency. Now, if it was accurate, then this would be purely linear, right? This would just keep going in a linear fashion. You'd have a linear slope, slope of 1. But it turns out that as the actual map distance increases, the recombination frequency tends to like level off, right? And so that means that it's going to underestimate the actual map distance. So when you start off, you're good, right? Like 10 is 10, right? So 10 as a recombination frequency is 10 as your actual map distance. Then when you get out to like, say, for example, your actual map distance is 60, well, you're in the what is that? Let's say 38%, right? Something like that. I don't know. But the point is it's going to underestimate that. Okay, so that is linkage.